Okay, welcome everyone to the TESS seminar for this week. We're extremely lucky to have Associate Professor Katarina Sam join us from Czechoslovakia, Czech Republic. So Katarina received her PhD in 2013 from University of South Bohemia, and she's now Associate Professor there and heads the Laboratory of Multi-Trophic Interactions. And so Katarina will discuss predator control of herbivores and has been conducting a really fascinating experiment over elevation and latitudinal gradients. And one of her sites is the Daintree Rainforest Observatory. And so I'm really looking forward to hearing about the projects. Thank you very much, Katarina, for joining us and for getting up early and for taking time out of your busy day.
Yeah, thank you very much. So I'm actually greeting you from Poland, not from Czech Republic. I arrived here yesterday, so I hope that everything with my internet will be okay. And as it was announced, I really am going to talk about the predators and a bit about the birds, and only a little bit, unfortunately, about the results from draw, which we are still working on. But to start with, you might be aware that the one hypothesis which is generally defining why our world is green is the bottom-up control hypothesis, which is saying that the plants themselves are so disgusting and so producing so much of the biomass that they themselves protect the biomass and the green foliage against the herbivores. This hypothesis is the original one, the older one, and only later on in the, this is generally saying that the plants have several types of defenses which they co-evolved during the very long evolutionary race between the plants and the herbivores. So the plants develop the constitutive defenses which are always present in the plants, and thus they might be useful in the situations where the arthropods, herbivorous arthropods are rich, abundant, and they are attacking the plants repeatedly. On the other hand, the induced defenses are only started and induced after the attack of the herbivores. Thus they might be useful in the situations where the arthropods are not so abundant, maybe in the cooler, less productive habitats, and where also the arthropod attacks do not occur repeatedly. So for tropical cairns, I would expect most of the plants to have constitutive defenses and protect themselves effectively. But there is also the other way how to protect the biomass of plants, and this is the top-down control hypothesis. This hypothesis was established in 1960 only. And at the beginning, it was quite controversial because it was saying it's not about the plants, but it's about the predators who are eating all the arthropods, thus letting the plants to flourish. Today, we know that the truth is somewhere in between. And even in the top-down control hypothesis, the plants are playing their role. This is because when I was mentioning the induced defenses can be split into the direct ones and indirect ones. The direct ones are aimed against the arthropod herbivores, killing them, making them very sick, or indirectly, when the plant is eaten by herbivores, the plant is producing either visual or very often chemical signals which are perceived by the predators or maybe parasitoids, and they come and eat the herbivorous arthropods. So this is the top-down and bottom-up control, which are very likely in the interplay of what is happening in the ecological systems. But when we are talking about the predators, the question is what type of predators? The hypothesis was not specific, but many people, of course, assume that the insectivorous birds as the big predators can be very effective. But when we look at their biomass, it's actually very tiny globally. Thus, ant people very often claim that the ants are the most important predators in many ecosystems. And there are also studies showing that spiders might be very effective because they have got relatively big biomass, and they eat actually similar amount of tons of the insect annually. Actually, the number estimate is around 500 to 800 million tons of insect for birds and 400 to 800 million tons for spiders. So what are the important predators in which system? And the follow-up question might be where the cascades from predators down to plants actually occur. The traditional view is that the terrestrial trophic cascades are strongest, is the most productive sites. And the Oxanen's hypothesis actually was saying that the top-down control varies as the function of productivity. But the follow-up reviews didn't confirm this. They actually found the trophic cascades occur in the wet, at the extreme most wet and the driest regions. And the other review found the strong trophic cascades to occur only in the temperate regions. So why is it so difficult actually to study the predation and say where the predation occurs and where the trophic cascades from predators to plants occur? That's because the predation is happening really somewhere above your head, typically very fast, and very often it's difficult to find what the predator ate, how much of it they ate, because we can't follow them in the time. So there is several indirect methods how to study predation. The one focuses only simply on the predation by exposing the baits. This can be a real insect, that insect like larvae, which you pin to the foliage, and then you can count how many of them was missing. Unfortunately, this method doesn't reveal what was the predator unless you set many cameras to observe them. The other method uses the plasticine or some other paste, which can keep the imprints of the predators, and from the baits you can identify what kind of predator was trying to attack and take the prey. Of course, this method is less realistic. The dummy insect doesn't look exactly the way as it would look. It doesn't behave anyway, and it has also different smell. So the third method, not like third, which I am involved in, is the exclusion of predators. This is done by the cages or maybe some other barriers, and it actually exclude the predators, and after that we can analyze what communities of insects are kept in the cage, thus not eaten by the predators, and we can exclude this way birds, bats, vertebrate predators. At some studies they were doing the exclusion of lizards, but also you can use some sticky barriers to exclude ants. But one of my questions was whether these methods actually provide similar results. I was lucky to be involved in a study which exposed the plasticine caterpillars across the globe. And in this study, I was working in Czech Republic, Brisbane at the time, and in Papua New Guinea. And we were gluing the caterpillars on the trees. And we actually found that the highest proportion of the caterpillars was attacked by the arthropods. And of course, many of them was attacked in the tropical areas. As for the birds, there was no trend along the latitudinal gradient, neither along the elevational gradient. And overall, the predation by the birds was very low. But still, I was not very happy with the result because I really believe that the birds might be eating more. And it's in simply the methodological aspect which we are missing here. And because I found out that there is actually many exclusion experiments already existing globally, I reviewed them and I focused on what happens in the literature, based on the literature, when the vertebrate predators and ants are excluded from the study system. Here, where we are looking at the caterpillar graph, you can see that after the vertebrates, predators are missing from the study system, the total arthropod abundances increase, usually more than 2 times globally. But when the ants are missing, the effect was not significant. At some study sites, of course, the other arthropods increase their abundances, but the effect didn't differ from 0. Quite interesting was the look into the predatory, into the arthropod gills. Looking at the predatory and herbivore arthropods, we found the difference in how ants and the vertebrate predators affect them. While the birds' vertebrate predator seems to be feeding on both gills in the equal proportion, ants seem to be preferring feeding on the herbivore arthropods only, and they didn't affect other predatory arthropods. But, and this actually transferred quite well and strongly to the planted image as the lower trophic cascade. When the ants were missing, the herbivory increased significantly because the arthropods which were not eaten by ants had higher chances to survive. But the effect of the birds was kind of melted down and diluted by the fact that they were acting as the intragill predators, and they were eating similar proportions of predatory and herbivore arthropods. Along the elevational gradient and latitudinal gradient, we again didn't find any strong effects. Like there is a cloud of dots, then statistically there is some trend, but I don't see actually real result there. Along the latitudinal gradient, it's more similar to what we have seen from the plasticine caterpillars, higher effect of the ants on the herbivory in lower latitudes. And interesting thing which we found in the review is that the experimental results are affected by the duration of the experiment. Especially for the exclusion of the ants, the effect is stronger when the ants are missing for a very short time, while the abundances of arthropods actually decrease when the experiment is long, approaching 1 year. And because many of these results were also very difficult to compare because the methods were different, duration of the experiments were different, we decided that we really need to do experiment which is spanning several study sites and uses standardized methodology. So this is where I started to do the exposure experiments. For the first time, I did it in Papua New Guinea along the elevational gradient of Mount Wilhelm, which is the highest mountain of Papua. And I was working along the elevational gradient also before doing my PhD. And this elevational gradient spans from 200 meters to 3,007 meters above sea level. And it has got 8 more or less permanent study sites, camps where I could have been based. The elevational gradient, of course, differs a lot. Each of the study sites have very different habitat conditions. In the understory, the forest is very high, very dense and dark in the understory, while towards the top of the mountain, the canopy starts to get open. The understory is very thick, and the trees are about 3 times lower, from 30 meters decreasing to 10 meters tall trees. And also the temperature and the humidity changes significantly along the elevation gradient. With the lowlands, we had a mean temperature 25 degrees, while at the top of the mountain, we were facing the morning frosts and mean daily temperature only 7 degrees. The humidity was more or less stable, but all this resulted into the very different communities of predators which we were dealing with. We were surveying them quite intensely. We were focusing on the ants, which we were surveying by different weights, and also on birds, which I was doing by myself with my assistant, Bonnie Kwane. And we were surveying by point counts, by misnetting, what actually lives there. And as the additional thing, we were making the birds to vomit. We were giving them very bitter emetic tartar, and in these bowls, we made them vomit, and we were taking the samples and identifying what exactly they are eating. One of my students, Peter Amick, was also surveying the bat communities. And unfortunately, something happened with the samples. We could not sequence properly due to the storage problems of what the bats eat, but we are still catching up and doing this now later. And after I knew what is actually happening with the predator communities, and I found that the ants are, for example, not present above 2007 meters above sea level, I started to do the exclusion of the predators. I have got saplings in the forest understory to which I didn't do anything. I just marked the leaves on them and let them be by themselves. Then at some saplings, I excluded the ants by sticky barrier, by tangle glue. And then I had a treatment where we excluded vertebrate predators by cages which were placed permanently around the saplings, and the combined treatments, exclusion of the birds, bed, and ants together, just having the cage and the glue all around and on the trunk. And then those two tricky treatments, which we were doing only for 1 month, and then we were having the cages which we pulled up in the morning and in the afternoon to let the respective predator in. So all the other treatments we kept for 6 months and then again 6 months during the wet and dry season, but those bird and bed treatments, due to the really intense work on them, we kept only 2 times for a month. And this is how it little bit looked like in the field, my sketches of the treatments. And then we used the water pipes, which we purchased and carried to the forest, and the assistants in Papua building the exclosures along the saplings. After the 6 months, we surveyed the complete arthropod communities. Here we were wrapping the crowns of the saplings to the mosquito net, killing the insect with insecticide and catching generally everything which we found. And we also collected 30 leaves from each sapling. And at the end of the experiment, after 1 year, we collected all the leaves from the complete sapling. At the beginning, we of course had to number the leaves to mark them, so we know how the herbivory changes over the time. This was a bit challenge, both because the permanent markers do not last full year, and also because it's very analytically demanding to outline then all the herbivorous damage from all leaves. But in Papua, we finished all the work. And these are the results. Here we are looking at the effect of the predator exclusion along the elevational gradient and how the arthropod abundances actually changed. So in the black line, you can see the control, how the arthropod abundances per meter square looked like on the control saplings. And then in the blue line, you can see that the arthropod abundances increased a little bit, but not significantly, when the ants were gone. But significant difference was detected only in the exclusion of all predators and the vertebrates. Thus, in the cages, the arthropod communities increased significantly. The effect was stronger in the wet season than in the dry season. During the dry season, there was generally less arthropods altogether in all treatments. And when we were looking into which guilds and the groups of the arthropods were the most affected, we have seen that the spiders were significantly increasing their abundances, but surprisingly, only when the ants were missing. Especially when the ants were missing, which means that in the treatments where all predators or ants only were missing, the spiders increased their abundances significantly, around like 2 times. And the other groups of the predators, arthropods, were also affected, mostly the all other predators, Orthoptera, Lepidoptera, but some of them not significantly from 0. And this is in line, actually, this is only the way, different way how we can look at the results. Here is the proportion of the spiders in the sample. So proportion of the spiders towards the other arthropods in the sample. And the gray space is the control. So, and here is about 50% of the spiders in the sample. So in control treatments and the vertebrate treatments, it didn't differ too much from the control, from 50%, but especially in the lowest elevations, when the ants were missing, the spiders increased their abundances significantly, which means that after a year after 6 months and 6 months again of the experiment, the spiders took over the free niches which we made by excluding the ants. And this might be the reason why the long-term experiments maybe didn't find any effect of the ants exclusion, because the spiders obviously needed some time to get in, and it didn't happen immediately. In the shorter experiments, there was not change in the proportion of the predators versus the other arthropods, but it was happening only over the time. So as long you have the experiment longer, the spiders are finding the niches more effectively, getting in and increasing their abundances, thus again melting down the effect of the ant exclusion. And when we were looking to the effect of the birds, we compared what we found with the data which we analyzed from the diets taken by birds. It's quite messy, I have to say, but in yellow you can see that the birds are actually taking quite many spiders, typically, and quite many beetles, which is in line what we have seen before. The beetles and spiders were the most affected by the birds. This method is actually showing the visual identification compared with the metabarcoding, and there are also some discrepancies in the methods, what they can reveal and what they can't. But overall, the beetles and the spiders are the most eaten items by birds, and these are the most affected by the exposures of these predators. You might seen that I didn't talk about the bird and bat exposures so far. That's because the results are quite messy. I think we would need to do the experiments for much longer time, but we found that the effect of these two types of predators is definitely not additive. But at some of the study sites, the birds seem to be more important, and other sites, the bats seem to be more important. And overall, I think we would really need to increase it for a longer time. Because not all the bats are eating the insect, some of them are frugivores, some of them are mixed feeders. We are still working out what specifically they eat, and we are getting new samples and trying to match the data together. When we look to the lower trophic level, to the herbivory, the exclusion of predators had again a very similar result effect on the herbivory. Again, when the control trees are shown in the black, there was no change in the herbivory, or very tiny change in the herbivory after the ants were missing. And this is what I was talking before. That's because very likely the spiders took their space after the 6 months and filled in the niches and started to act as the predators instead of them. But when the vertebrate and all predators, which means again vertebrates and ants, were missing, the herbivores damage increased significantly, but just by 1 or 2% in the lowlands, which is way lower change than I expected. But still, this level of herbivory might be ecologically important for the plants, because overall global herbivory is estimated to be around 2 to 3%. Some studies estimated the global standing herbivory only as 1%. And Kozlov in his review found the highest herbivory in the temperate, and overall global average 7.6%. So the increase from roughly 3% to 5% in the tropical forests in Papua is not a big number. It's not a big change as a number, but it might be really important for the plants. because the other studies which I found that the 5% herbivory actually transfer into the 15 to 25 decrease of productivity of the plants. And Schlinkert also showed that 1% of herbivory translate to the 10% lower abundance of fruits in the future years. And as we were dealing with the understory saplings in the very dense and dark tropical understory, these saplings are very dependent on the light, and every bite counts for them because the missing herbivory might affect them significantly and actually kill them. The other things which we found is that the results are very much dependent on the plant species which we are working on. Here, I would like to show the case study of the Ficus alliana, where the herbivory, natural herbivory, standing herbivory on the control trees decreases with the increasing elevation. We also found that in this plant species, the constitutive defenses, the protection which is always present in the plant species, increases with the increasing elevation. And we were testing the inducibility of the plants, and we found that the inducibility is actually higher in the lowlands, which means that the plant species seems to be more dependent on the help of the predators and calling for the help in the lowlands, but it's protecting itself and the more expensive biomass in the higher elevations. So based on this, we expected, and we also found that there is a big difference between the control and exclusion treatments in the lowlands than in the higher elevation, which means that when we removed the predators, the plant was calling for help more in the lowlands, but could not reach the predators anymore. Thus, the herbivores had higher chances to eat them effectively. This was not happening too much in the higher elevation. So based on these results, I would like to kind of conclude in the middle that there is a very complex interplay between the top-down and bottom-up control, which works on the basis of the characteristics of individual plant species. I showed you the example of Ficus halliana, but there is many more plant species for which we found similar results. So depending on where each plant invests in, whether more into the induced defenses or constitutive defenses or equally into both, it affects how the top-down control will be important for it. So overall, at one study site, it really depends which plant species you are working on and how the composition of the plant is actually there. We found that the vertebrates overall, and especially maybe birds in lowland forests, are the most effective predators of the arthropods, yet the cascades from birds to plants are always weakened by the integral predation, hence are effectively feeding on the herbivores. Thus, under the normal conditions, they would have very strong effect on the herbivory, but it seems to be depending on the length of the experiment. And overall, over some time, the effect seems to be diluted by spiders or other mesopredators, which are getting in instead of ants. And because all the other experiments which I was doing so far were conducted in the forest understory, I wanted to find out what is actually happening above our heads in the forest canopies, where the trophic cascade seems to be stronger. There is many more predators, definitely more foliage up in the canopy, and more light, and there is a higher productivity in the canopies. So how it looks like there. For that, I needed to use the canopy cranes, which got me up with the same treatments as I was doing. Unfortunately, at some of the study sites, crane sites, we were not able to do the bird and bat exclosures in the canopy, but we did them always in the understory. And we generally replicate the same experiment in slightly bigger scale at, we aimed for 6 study sites along the latitudinal gradient. So we so far finished the work in Tomakomai, where we spent 2 years, 2 seasons. We finished the samples in Cairns and in Uke Face close to Sydney. And we have finished 3/4 of the experiment in Papua New Guinea. We are going to do the final sampling in January of next year. And we also finished the work in China. Unfortunately, we are not allowed to ship the insect out. So we are a bit struggling there, but we have got the data on the Herbivory and we hope to see our insects ever again. And our final study site was supposed to be the Lambert Hills in Malaysia, but unfortunately the canopy train was shut down and closed last year and we can't return there anymore. So just to do something and keep ourselves busy, we moved our study site, the last study site, to Leipzig in Germany, where we started to do the experiment last year. Actually, this is quite unexpected move because we are moving away from the coastal rim of the Pacific, but Leipzig has a very similar plant composition as the Tomakomai. It's slightly higher. And as you will see, there is very strong difference between the temperate and tropical study sites. So to have more or less temperate third study site together with Tomakomai and Uke Face was more natural move than trying to have the fourth tropical study site. At each of the study sites, we worked on phylogenetically pre-selected plant species, but we of course had to adjust the selection to the abundances because we had to reach them from the canopy train. But we try to standardize the plant defenses, and we are also analyzing various types of the plant defenses from the selected plant species. And the project is quite long. At each of the study sites, we are working with 720 saplings and 150 branches in the canopy, and we used many PVC pipes, lots of netting, and we so far analyzed and identified several hundreds of thousands of insects. This is what my students are doing now in the laboratory. And we again try to survey the predation by the plasticine caterpillars method. And here are the results, which are actually quite surprising to me and very unexpected. Oh, now I can see that there is actually the Leipzig data missing, but I can tell you that the results are very similar to what we see here in Tomakomai. So in Tomakomai and Uke Face and in Leipzig, we found the predation to be higher in the canopy than in the understory. And in the tropical study sites, in Bubeng, Kakoba, and in Cairns, we didn't find that effect, and it was more or less opposite, higher predation, if it was significantly higher in the forest understory than in the canopy. This seems to be not very well matching the abundances and biomass of the predators. Here, I am showing you the results from the entry. There is a arthropod predation because arthropods were responsible for most of the predation at the plasticine caterpillars. There was higher predation by arthropods in the understory than in the canopy. And the bird predation overall was very tiny, 10 times smaller than the predation by arthropods, and quite similar between the canopy and understory. This correlated well with the distribution of the abundances of ants. There was more ants found in the understory than in the canopy in the entry. But for the birds, we found much higher biomass of the birds and higher number of birds in the forest canopy than in the understory. But then when we subsampled them to the insectivores, there was actually more insectivores in the understory than in the canopy. But again, it didn't match well the results from the plasticine caterpillars. But let's see how it will affect the results through the exclusion of the experiments, because you might already see that the plasticine method doesn't always reflect what is actually happening in the study system. So the results are actually very, very exploratory here. I was getting the data only 2 days ago and drawing the graph yesterday for day 3. But overall, it seems that the invertebrate abundances were slightly higher, or at least there was a higher variability in the canopy than in the understory. In the light blue, you can see the control trees, which had always quite low abundances of arthropods, but after the predators were missing, the abundances increased. In the canopy, they increased very much. I think it might be significant after we ran proper models, but they increased about 2 times after the vertebrate predators were missing, and they slightly increased also when the ants were missing. In the understory, the trend is similar. Again, when the vertebrates were missing, all predators were missing, they increased. But similarly as in our earlier experiment, the simple treatment, bird and bat exclosure, didn't provide any conclusive results. I'm really wondering what will happen after we put all the factors to the model and run it properly. But as far as I'm talking to you today, I just wanted to show you what we specifically found in the day 3. As we finished the work in Tomakomai a bit earlier, we are done there with proper counts, and here are the final results and the models. In the understory, similarly as in the day 3, the herbivore abundances were slightly lower, and we found no effect of the exclusion, but after the birds and bats were excluded, the abundances increased significantly, more when the birds were missing than when the bats were missing in the understory. When we combined the treatments, excluded all predators together, it was still significant, but a little bit weird things were happening there. But still significant increase in the arthropod abundances. To our surprise, in the canopy, the increase of the abundances of arthropods were crazy high. This is the control in light gray. And when the vertebrates and all predators were missing, the abundances increased from 10 individuals per meter square to 200 individuals per meter square of foliage, which was absolutely crazy. And this is not what we observed in day 3, definitely not. In Tomakomai, the biggest variability in the, this time, herbivory per 3 individual was explained by the strata. There was always higher herbivory in forest understory than in the canopy. And you can see that the plant species was important factor in the analysis. There were plant species which were eaten a lot, like Ostraya or Betula, and there were plant species which were not eaten too much, like Magnolia. But again, the exclusion of the predators had significant effect on the herbivory. It always increased when the predators were missing in all treatment and where treatment. So where the cages were there, the abundances of arthropods increased significantly, typically by 60 or between 60 to 100%. So we expect quite similar results for day 3 and other study sites. I actually believe that the vertical stratification will be not so big for the tropical study sites, but we will see differences in the importance of predators for the temperate study sites, but this is yet to be analyzed. And with this, I would like to acknowledge all my students who were working hard and identifying all the arthropods and mainly analyzing the herbivory from the photos, from the scans which obtained at each of the study sites. And thanks due to the very, very difficult COVID times where we were not able to get to the study sites, the big thanks goes to all the technicians and assistants, to Michelle, Vinod, Craig, and others who saved our experiment where we were not able to get to Australia and do the final collections by ourselves. And as you might see that the experiment is still under the way. If you wish to learn more and follow our up-to-date results, you can follow us on the Twitter. And thank you very much for attention, and I'm happy to take any questions you might have.